Hey everybody, Mr. Murphy here. I'm going to talk about chapter 3.4, which is all about regulation of enzymes. Remember, enzymes are catalysts. They are facilitating chemical reactions, and therefore they're a very important part of the broad metabolism going on inside of a organism. And it turns out that there's a lot of control involved in that because we don't really want our enzymes just freely facilitating the breakdown or buildup of like everything they can find. So it should be kind of logical that we have mechanisms by which we can turn on or off enzymes beyond just like how many we're making or things like that inside of a cell. So this is very much tied into the idea of homeostasis. So it's a good opportunity just to remind us that homeostasis is the maintenance of stable conditions inside of a cell. Whether it's amount of water or glucose or the temperature or the pH or anything. It is creating a constant environment that is ideal for the evolution of the parts inside, that the shape of proteins and things like that are dependent upon specific conditions inside of the cell, and so the cell wants to maintain that. That maintenance requires careful control over what types of chemical reactions are going on inside of the cell, because if we had just a runaway enzyme that's just like breaking down all the things, then you're going to get a buildup of too much of something, which too much of anything becomes problematic inside of the cell. I mean, you can die from too much water. There's literally no chemical that can't become problematic if there is too much or too little of it inside of a cell. So cells have the ability to turn the synthesis of enzymes on and off. So that is to say, like the transcription translation process of taking DNA to RNA and then building new enzymes, that can be turned on and off. We'll learn in our next unit how the cell, because of lysosomes and other functional components inside of the cell, it's constantly breaking things down. So our cell's constantly working to like lower the amount of things, which is kind of nice. It sounds bad, but it's kind of nice because then we can control how much we're making in response, and it gives us like a way to control. So simply by making more enzymes, we can control it. But there are also inhibitors and activators that we can use to sort of turn on or off enzymes as well. So that way we don't have to worry about like how many enzymes do we have. We can have all the enzymes we want, and we can instead then create or deliver more or fewer inhibitors to the enzymes in order to control their activity. There is a type of inhibition that can be irreversible, and this is where there's a covalent interaction between the enzyme and the inhibitor, and this would permanently shut down the enzyme. So like all things, lots of examples that utilize different mechanisms for sort of the same general idea of inhibiting or activating enzymes. So here's an example of acetylcholine esterase, which is an enzyme, and it has a particular active site, and an inhibitor can come in and bind to that active site and shut down the acetylcholine esterase enzyme. You don't need to know that word. You don't need to know this example, but the idea here is that it's covalently bonding and therefore irreversibly shutting it down. So it's just an example of how that can happen. There's two broad types of inhibitors. There's ones that are called competitive, and they're called that because they compete with the substrate to bind to the active site. So this would be a competitive inhibitor. It is binding in the active site. So it has a shape that's attracted to the active site. In this case, though, it then leads to a covalent bond and shuts down the enzyme. But other types of reversible inhibition would just be things that fit into the active site, just like the substrate does, but it's a different molecule than the substrate, so it doesn't get broken down or whatever. It just sort of gums up the works and slows down the enzyme. Again, sounds bad, but there's times when we just want to slow down our enzymes. We don't want them to work as hard. A non-competitive inhibitor is one that binds somewhere else on the enzyme. And so this would be if the active site is here, it's grabbing on somewhere else and changing the shape. And there's some diagrams to show that. So competitive inhibition, it competes with the active site, blocks it, so the substrate just can't get in there. Non-competitive, the inhibitor binds to a different site that's called an allosteric site. Just to give you that vocab term to recognize, an allosteric site is just somewhere else on the protein, on the enzyme protein, that when that little green bean thing is nearby the enzyme, it causes a shape change in the enzyme. not through any fancy mechanism. Just this has a chemical profile, a set of positive, negative, hydrogen bond, whatever, all that sort of normal shape stuff. And when it is glommed onto the side of our enzyme, it changes the shape of the constituent amino acids inside of that enzyme and can lead to the active site no longer fitting the substrate. It's not, this Venus flytrap example is kind of oversimplifying it. It's realistically, when you get that allosteric thing, it's just causing some sort of twisting or folding or change to the shape. It doesn't have to be like, nope, it's fully wrapped up like a dumpling and now it doesn't fit the substrate. It's a good way of thinking about it, though. So that's allosteric regulation. So it's the non-substrate molecule, which is a fancy way of saying not the thing that's supposed to be acted upon by the enzyme, and it binds somewhere other than the active site. Changes the enzyme shape, which means that it shuts down the enzyme. Now, it can activate it. So if we think about this like in reverse, if maybe normally the enzyme's closed, but when the non-competitive molecule here, which would be considered an activator in this example, maybe when it comes in, it causes the active site to actually open up. So it could go either way. Either it changes the shape of the enzyme in such a way that it prevents the enzyme from working, or maybe the enzyme started out with a bad shape and this activator now changes it in order to activate it. I said bad shape. It's not that it's bad. If something requires an activator, it's sort of in a generally turned off state, which is good because maybe we don't want it activated. And then when we want it activated, we generate the activators that kind of are the key to turn on the enzyme and to allow it to do its job. So there's a lot of good diagrams that sort of show the way in which you could have an activator enzyme, or you could have a inhibitor, just a way in which we can use allosteric sites to open or close the active site to the reactants. Protein kinases are a term that we're going to see a lot in our next unit. These are enzymes that regulate responses to the environment. So a protein kinase is just a really broad term for this is a enzyme which is facilitating a response to external signals or the environment or whatever. So these are like mediator enzymes that are facilitating the change from one molecule to another. And what we'll see is that really there's this cascade of many different kinases all working together to lead to one end result. This will be our first introduction to a pretty important term, which is feedback inhibition. Really simple. It's just where the end product of a chain of events is the thing that inhibits earlier steps. Super handy control mechanism inside of cells that we can inhibit the production of something because the product that we're making is the inhibitor. So having sort of a particular shape to enzymes that allows, if we're making a bunch of, let's say, I don't know, if we're making a lot of glucose, whatever, you're a plant cell, you're making a lot of glucose. And as that glucose level climbs, you don't want to keep making glucose. Well, if glucose sort of becomes an inhibitor to an earlier step, then as the concentration of glucose goes up, the active nature of that enzyme that was sort of an integral part in that chain goes down as the glucose inhibits it more and more and more, which the end result is we don't make as much glucose, which is good because we already have a lot of glucose. Then as the glucose level maybe gets used up, suddenly there's not as much of an inhibitor and now you have more activation and the process goes on. So here we can see the final product, in this case isoleucine, inhibits this step here, converting threonine to ketobutyrate. You don't need to know these words. I don't know these words. The point is that this is feedback inhibition because the final product is somehow inhibiting an earlier step. Good control mechanism. We talked about how pH can affect enzyme activity more broadly in like a protein sense. So because these different side chains can be attracted or repelled by H pluses or OH minuses, that can affect the shape of the protein, which in the case of enzymes can really alter their functionality. generally for the worse, because most places in our body we maintain constant pH. But there are examples where this could actually be a plus, and one key example would be in our stomachs, where the generation of pepsin, which is a protein that breaks down, or it's an enzyme that breaks down proteins in our stomach, we don't want that always active. Because if your stomach's empty, there's lots of other proteins that could bump into and annihilate, like proteins inside of the cell membranes of the lining of your stomach. So you don't really want that to happen generally. And so the idea here is that the pH can actually instill a change in the shape and environment of the pepsin in order to activate it, so that when you've begun eating, when there's increased acid being poured into your stomach because of brain signals and things like that from the response to you consuming food, then the pepsin becomes activated. So there's all of the things that we've been talking about can be leveraged in a positive or negative way, just sort of depending upon the conditions and what's actually going on. So this little section is just sort of a reminder of that these influences that we've talked about influencing the shape of proteins applies to enzymes as well. So temperature, same deal. We have optimal sort of activity at certain pHs, optimal activity at certain temperatures. Just going back to the pH thing here, you can see that pepsin is optimal at a pH of 2, which is that hydrochloric acid environment of the stomach, whereas most other enzymes we find inside of our cells are going to be focused somewhere around a pH of 7. Depending on the organism, they may be more basic or less. Temperature, same thing. You have an optimal rate. Lower, and there's denaturing, and same thing, higher, denaturing. And you notice on these, the temperature line usually ends at a certain point, because beyond that point you have irreversibly denatured it. If you boil an enzyme, that's enough heat energy, it's going to tear apart those covalent bonds, and you're not going to come back from that. So at a certain point you just drop off and have no activity. But do notice that, it's not just like perfect temperature, everything's good, and anything other than that is zero activity. The enzymes still function. It's just how challenged is that shape by the environment. One last thing to talk about with enzymes is the fact that some organisms have what are called isozymes, which is just a fancy word for enzymes that do the same thing but have different shapes. And from an evolutionary standpoint, that's going to be really beneficial, because that's going to be an organism that has greater hardiness to change in its environment. That if it has an environment that changes in one way that shuts down one version of that enzyme, hopefully it's got backups that still operate, function well within the new sort of range of what the environment is, whether that's pH or temperature or whatever. So there's an evolutionary component to this as well, which is important for us to remember in the background, because evolution is sort of the driving mechanism for why things are the way they are today. And it's important for us to understand these examples as just good reminders that, yep, there is sort of contingency plans in life, and that's what's allowed, our species to make it till today, billions of years into the process of life. All living organisms today have a variety of ways in which they're able to, respond to changes in their environment, some better than others, but it's got us to where we are. Cool. So the end of this chapter returns to that aspirin idea from the very beginning, which was sort of asking how do proteins and enzymes sort of explain the shape of aspirin. Aspirin is something we're probably all familiar with. You take an aspirin if you have a headache or you're sore or whatever, aspirin, Tylenol, ibuprofen. It's all very similar in function. But it's a chemical, and it has a particular shape. And in the case of aspirin, it inhibits a very specific enzyme. So the shape of that aspirin is matching the shape of a particular protein, a particular enzyme. And this is an enzyme that catalyzes a specific metabolic pathway that leads to molecules that lead to inflammation. So these are anti-inflammatory drugs that then reduce the inflammation, which then can relieve things related to inflammation, pressure and things like that, and pressure in your sinuses and your brain, sore muscles, all of that. And so it inhibits that enzyme because it has a very particular shape that matches that enzyme and can can prevent the enzyme from being able to produce more of these molecules associated with inflammation. But it's not like you take an aspirin and just all production of prostaglandin is gone, right? That'd be bad. We don't want permanent shutdown, and we don't want complete shutdown ever of things that are going on inside of our body. And that's why concentration is such an important thing. And you might take 1 aspirin, or you take 2 aspirin, but you're not going to take 12 aspirin. That can become very problematic because too much of anything is going to start to have unintended consequences. Either that the concentration of it becomes so large it becomes problematic elsewhere in the body, or that you've just shut this down too much. This is a natural process, and it's a molecule that we need inside of our body. And so that's why it's sort of careful balance of these things is so important, because we don't want to throw off the homeostasis of our body too much. But sometimes we do like to step in artificially and be like, I don't want my head to hurt anymore, I'm going to take an aspirin. So that's just a sort of real world connection to why protein shape matters, and why when developing new types of drugs or treatments for things, it's important for us to understand the shape of these molecules in order to design or synthesize or whatever new molecules that can facilitate whatever action we want, so we can reach in and sort of control the activity of that enzyme. And that's the game of all medicine, really, is about finding things that work. And for much of medical history, that's just like, oh, I ate this plant and now like my arthritis is gone. Today it's a little more nuanced, and we can actually understand molecular structures, and we can actually generate things that are not naturally found in the world, but chemically can exist in order to target very specific enzymes and treat all sorts of illnesses, genetic disorders, cancers, all sorts of things. So it's cool, but it's scary how much chemistry is actually involved. So that is chapter 3, which is a big one. If you have questions, let me know.